Hello and good evening once again.
Hello and good evening.
So we have already discussed, or particularly I have already discussed Unit 3, 10, and today I have come up with Unit 11, mostly Unit 11 Part E. Because you see in Unit 11, which is your evolution, you know, uh, here Part A, B, C, and D. Unit 11, Part A, B, C, and D, they are absolutely theory based. Okay? You just have to memorize those experiments, uh, the era and the period and the examples and what the scientists said and all. Unit Part E, which is the population ecology, this is theory plus numericals. For every theoretical concept, you have numericals except for the speciation part. Okay, under this sub topic E, you also have speciation, but speciation is doesn't have any numericals. Instead, it just have the theory. Okay, if you go into the part F, which is your brain and behavior, there also you have the theories, experiments, and all, and there is a numerical only from one section, that is the kin selection. Okay, only from this part you get numericals, otherwise not. Otherwise it's mostly theoretical. So maximum numericals comes from your population ecology, sorry, the population genetics. So today I thought, why not discuss the numericals? Okay, because theory is, if you are clear with the concept, you will be able to answer the theoretical question. At least you're expected to answer. Okay, but many a times what happens in the numerical part, students are stuck. Okay, or maybe due to some careless mistake they have gone wrong. And the best part of the numerical is you just have two formulas here, two to three formulas here, not much. Okay, how to calculate the genotypic frequencies, that you have to study, how to calculate the allelic frequency, that you have to remember, and how to calculate what is the Hardy-Weinberg equation. So p square plus 2pq plus q square equals to 1, and p plus q equals to 1. And after that you go into the derivations of the Hardy-Weinberg. Okay, so when you deviate from Hardy-Weinberg, then the deviate, when you deviate from Hardy-Weinberg, then what happens? Those calculations are there, right? This is the actual set of the calculations that you have to do. Yes, good evening, Malini. So again, like the other sessions, I have 10 numerical questions today, and I'll give you 2 minutes time for each of the questions for you to solve, and then I will solve it for you so you can cross check. Okay, so shall we start? Give me a thumbs up if everyone wants me to start, if everyone is ready to start the session. Or you can type BIO, bring it on. Let's see how many of you give me this. If you want me to start the session, if you're eager to solve the questions, type BIO, bring it on. Type BIO in the chat box. Okay, I see Malini is ready. What about others? Arzu is also ready. That's great. Others, what happened? Are you thinking, should I try the numericals or should I leave this entire section? Okay, Alka is also saying BIO. Okay, fine, great. So let's just start, okay? So the first question I have here is a very simple question. In a sample from a population where 65 individuals with BB genotype, 30 individuals with heterozygous B genotype, and 15 individuals with recessive B genotype, the frequency of the B allele is. The frequency of the B allele is what? So I'll give you 2 minutes time. So remember, you have to find out the allelic frequency. Okay, I'll give you a hint here. You have to find out the allelic frequency of B. Now there were two ways to calculate the allelic frequency. One was from the genotypic data. One from the genotypic data, and the second one was from the genotype frequency. Okay, so look here what kind of data has been given to you. Accordingly select select the correct equation. Okay, for this one there is a different equation, for this one there is a different equation. I hope my audio is very clear to everyone. Okay, Manini is saying option 2. Fine, others, can I get some answers? Okay, Arzu is also saying option 2. So basically the correct answer here is option 2. Very, very good. So for those who are still wondering how to do this, see, over here what they have given you, the number of individuals have been given to you, right? So that means the genotypic data is given to you. Okay, the number, not the frequency. This is not provided. The genotype, the number of individuals of a particular genotype is given to you. So I will use a formula. Frequency of small b is equals to 2 into n of small b plus n of heterozygous b divided by 2n. I will use this particular formula. Okay, so you just have to put the data here. 2 into small b, what they have given you, it is 15, right? The number of individuals who are small b is 15 plus number of individuals who are heterozygous is 30, right? Divided by 2 into n means total. So total means it is a 65 plus 30 plus 15, which will give you 110. Okay, so 2 into 110. Right? Now, if you solve this, you will see that you are getting you're getting 60 by 220, which is equals to 0.27. Okay? Can you see the annotation, or should I change the color? Is it comfortable for you? Okay, let me use yellow. I guess that will be better. Clear? So this is your answer. Okay. Moving on to the next one. So a founder population has an heterozygous genotype with a frequency of 1 and number of individuals with either homozygous A or homozygous recessive A genotypes. no individuals of either homozygous dominant or homozygous recessive genotypes. With repeated self-fertilization, the frequency of homozygous A, heterozygous and homozygous recessive A after 3 generations will be what? After 3 generations will be what? So this is talking about inbreeding. Okay, this is talking about inbreeding. So as we know that inbreeding will reduce the number of my heterozygous and my homozygous number will increase, right? So since all the individuals here are having the same genotype, that is the heterozygous, okay, so self-fertilization will result into inbreeding, which is a form of your non-random breeding. So try this and then I will discuss. Again, 2 minutes time I'm giving it to you. Very interesting question, a part C question. So a hint is that inbreeding will reduce the heterozygous by half. Right? Okay. Let me solve this question for you. So basically, when you self a heterozygous, what do you get? You get four kappa 2 heterozygous A plus 4 small a. Sorry. 1 second. Just a minute. I'll do it in the other way. So you are crossing a heterozygous. Okay, I'll do it in the checkerboard way so that you are comfortable. So you have kappa small a, and small a, right? So you have 1 kappa plus 2 kappa small a plus small a, right? Now what happens is, so when the heterozygous reproduce, only half of the progeny will be heterozygous, like the parents, and the other half is becoming your homozygous dominant and recessive. So this means that in each generation of self-fertilization, the percentage of heterozygous decreases by 50%, that is half, right? So frequency of heterozygous in generation 0, that is at the start, is 1. Okay, after 3 generations it will be how much? The frequency of your heterozygous will become half to the power of 3, right, which is 1 by 8. Now frequency of kappa will be how much? It will be equal to the frequency of your homozygous recessive also, right? Which is going to be 1 minus 1 by 8 by 2. That is equals to 7 by 16. So the correct option here is option 2. I hope everyone has understood this. Right? So getting into very good, Marini. Option 2, yes. Now getting into question number 3. So question number 3, they are saying that in a population of 2000 individuals of a plant species, genetic difference at a single locus leads to different flower colors. The alleles are incompletely dominant. Okay, the population has 100 individuals with the genotype RR, that is white. 800 individuals with the genotype capital R small r, and the remaining genotype is your RR, red. Okay, so what is the frequency of small r? So I'll just quickly write it down for you what is given here. N is equals to 2000, right? And RR, number of individuals who are RR is 100. Capital R small r is equals to 800. Cap r is equals to the rest, that is your 2000 minus 800. Sorry, 800 plus 100, this will be 900. Okay, which is equals to obviously your 1100. All right, and they have asked you to find out the frequency of R. What is the, this is a question mark. So how to do this? Again, they have given you the genotypes, the number of genotypes, number of individuals of a particular genotype. So I will use that previous formula, 2 into n of RR plus n of heterozygous divided by 2 into n. So that will give me 2 into 100 plus 800 divided by 2 into 2000. Okay, so if you do this, it will come as your 1000 by 4000, which is equals to 0.25. Okay, so the correct answer here is option 1. Yes, Malini, very good. Option 1 is the correct answer. Yes, Anusha. Absolutely, very good. Alka, yes, correct. Good, very good. Okay, moving to question number 4. Question number 4. So this is a deviation. This is based on a deviation from your Hardy-Weinberg, because Hardy-Weinberg says that there will be no mutation, right? But obviously in nature mutation is happening. You can't stop mutation. You can't restrict a mutation, right? So therefore, how will your Hardy-Weinberg frequency change when there is a mutation? Based on that, this question is there. So again, if you know the formula, you will be able to answer. This is again another type of question that you might get. So, what's the formula? So here they have told you the forward mutation rate A1 to A2. This is your forward mutation, which is your mu is equals to 1 into 10 to the power of minus 5. Okay. Reverse mutation A2 to A1. This is your new. This is equals to 2 into 10 to the power of minus 5. They have given you a lot of other informations also, like the frequency of A1, A2. But if you recall the formula, you will see that it is telling you that f of A1, it is going to be calculated as mu by mu plus nu. Why? Because F1 is nothing but P, frequency of P, of the dominant allele. Okay, frequency of dominant allele is written as P mu by nu plus mu, and frequency of the recessive allele is written as mu by mu plus nu. Okay, so I will not go into this because that is not required. This is what is required. So just put the values here. What is your nu? What is nu? 2 into 10 to the power of minus 5 divided by mu plus nu. So 1 into 10 to the power of minus 5 plus 2 into 10 to the power of minus 5. Okay. So what you can do is you can take 10 to the power of minus 5 as common in the denominator. So this will be 3 into 10 to the power of minus 5. Okay, so you cut this off. If you solve this 2 by 3, you will see you're getting as somewhat 0.66. So you can round it up to 0.67. Okay, so the correct answer here is option 3, 0.67. Are you all following? So far, if you have followed all the questions, give me a thumbs up. If you could understand all the question and how to solve them, give me a thumbs up. Let's see. Let's see how many of you will be able to solve these kinds of questions. So, okay, Malini has given me 2 thumbs up. Wow. Arzu also understood. Anusha, great. Others? Okay, if this is new to you all, then practice. I will tell you practice, okay? Now moving on to question number 5. Question number 5. See, this is also a CSIR question. All of these are CSIR questions, okay? Now try to solve this. Again, a very simple question, only the figures are huge here, okay? But still you will be able to do. Okay, Alka, Sri Lakshmi, great, okay, fine, that's great. So, this point is not required at all. For your question solving, only you need the genotype and the number. Oh, wow. Arzu is very fast to reply. That's great. Malini also, wow. Super fast. Super fast. So, everyone has got the answer as one. Okay, I'll do it. For those who are still working out, I'll do this. So very simple, they have asked for n, right? So again, the genotypic data is given to us. So therefore, I will use the same old formula. So I'm not writing the formula again. I'm straight away doing the calculation. Okay. So 2 into 1303 plus 3039 divided by 2 into 6129. Okay. So if you do this, you will get 5645 by 12258, and this will give you 0.4605. Okay, so answer 1. Okay, so again the announcement that George Best is going to start. By this time you already know what we are offering. Okay, all the advantages that you will get, and the batch is starting from 1st of July. All right, I'll continue with the question answer. Moving on to question number 6. 100 independent populations of Drosophila are established with 10 individuals in each population, of which one individual is of heterozygous genotype and the other 9 are of homozygous genotype. If random genetic drift is the only mechanism acting on these populations, then after a large number of generations, the expected number of population fixed for recessive allele is. So let's see who can solve this. Let's see who can solve this. Who's the first one to give me the answer? So again, 2 minutes time for all of you. Read the question, analyze the question. Leave out all the extras data. Just pick up the correct data. That is insignificant. How many number of generations? They have just given after a large number of generations. So what should be the frequency of smalling in infinite generation? Do you remember? Since they have not mentioned the particular generation number, they have just said after large number of generation. Do you remember what happens when there is inbreeding, when there is non-random mating and all? The heterozygous becomes a zero and the homozygous becomes half. Okay, so just see what is here. In each population of individual, in each population, so each population is having 10 individuals, and there is only one heterozygous and there are 9 homozygous, right? So even if genetic drift is happening, most likely 9 this homozygous are getting drifted away. Okay, so that means the homozygous inbreeding is taking place, right? So after a large number of generation in inbreeding, we know that the heterozygous becomes zero and homozygous becomes half. Okay, this is the structure of the population after large number of generation in an inbreeding population. Okay, and here since you have more number of your homozygous and heterozygous, and it's likely that the homozygous are getting drifted, and they will obviously while they are reproducing, they are going to show or exhibit the uh, non-random mating, right? Because there is no other genotypes here. So keeping this in mind, okay, so therefore I can say that the frequency of small a in infinite generation is equals to half or zero point 0.5, right? Therefore expected number of population is equals to 0.5 into 10, that is equals to 5. Okay, so the correct answer is D, 5. Populations fixed, number of population fixed for small a. Okay, that is zero point 0.5 into the total individuals in that population, which is 10. Okay, so the frequency becomes half, so frequency into the total will become the actual figure. So wasn't this a very tricky question? Do you find this a tricky question? But the concept is very simple. Have you noticed it? Tricky question, but the concept is very simple. You just have to have a very clear mind. You have to visualize what this question is saying. If this is happening, then what will be the next? If you can do that, if you have that clear concept, then these tricky questions also becomes very simple. Okay? All right. But I hope everyone understood how to think, how to, analyze step after step. One step links to the next step. That is very important. Analysis. How did I link the genetic drift with your inbreeding, and then the concept, and then how the allele is getting fixed? Did you see the step? That analysis is important. Okay. All right. Let's move to question number 7. This is a straightforward question again, but this time you have to be careful about the formula that you're using. A straightforward question, be careful about the formula you're using. So they have given you the frequency of the homozygous dominant genotype. So I'm taking that as A1. Thank you, Sri Lakshmi. I'm glad that you have found it easy to understand. Okay, quickly try the second one. We just have half an hour left, not even half an hour, 20 minutes, and we still have three more questions. Okay, Anusha is the first person to answer option 3. The correct answer is option 3. Very, very good. So, see, here again they are asking you to calculate the frequency of the two alleles, right? Frequency of A1 is how much, and frequency of A2 is how much. This is what they are asking, correct? But look at the data they have given you. They haven't given you the number of individuals of a particular genotype. Instead, they have given you the genotypic frequency. So this is your 0.16. Frequency of A2 is equals to 0.25. So from genotypic frequency, how to calculate your allelic frequency? The formula is, so let me do it for the first one. Frequency of A1 will be calculated as frequency of homozygous A1 plus half of the frequency of heterozygous, because see, in heterozygous you have A1, A2, both types of alleles, right? But you just need one allele, that is the A1. You're considering just the frequency of one allele. So that's why I'm taking a half of it. Now, once you know the formula, just put the figures. So this is 0.59 plus this is your half of 0.16. If you solve this, you will see you're getting it as 0.67. For the next one, frequency of your A2, the same way you do it. So I'm just writing the values. 0.25 plus half of 0.16. So if you solve this, you will see you're getting it as 0.33. Okay? All right. Moving to the next question. Question number 8. Again, a very simple question. Very simple question. So they are saying the frequencies of alleles kappa and small a in a population at Hardy-Weinberg equilibrium are zero point 0.7 and zero point 0.3 respectively. In a random sample of 250 individuals taken from the population, how many are expected to be heterozygotes? Okay. So Arzu, Malini, all of you are saying option C of 105. The correct answer is option C. Very good. But why am I not finding others answering? Only a handful of you are answering. What about the others? Only looking will not help. Only listening will not help in numericals. You have to try it yourself. Okay? Right or wrong, try it yourself. So I'm assuming that even though you are not responding in the chat, you are at least trying. Okay? If you think that I'm going too fast, well, I have to go too fast because there's a time limit here. But later on, you will obviously the recordings will be there in YouTube. So just go over and try the questions yourself. Don't look into the explanation. Try the question and then go into the explanation. Okay? All right. Let me solve this question. So see, they have given you frequency of A, which is equals to zero point 0.7. Frequency of small a, which is equals to 0.3. Okay? And n is given to you. And mind it, what they're asking. They're asking how many are expected. That means they're not asking for the frequency, but rather they're asking for the number. Okay? So when you have found out the frequency, you have to multiply it with the number to get the actual number. That means finding out the heterozygous is very simple. According to Hardy-Weinberg, it is 2p^, right? So you just have to put the value 2 into 0.7 into 0.3 and you will get the answer as your 0.42. Now, this is the frequency of the heterozygous. If you want to calculate how many are heterozygous, you have to multiply 0.42 into the total, which is your 250. This will give you the actual number of heterozygous in the population, which is your 105. Okay? So whenever they're asking for a number, always remember you have to multiply the frequency with the total. Okay, otherwise it is a wrong answer. Many a times they give you this trick. Okay, students just do the frequency and then they can't match the answer, they get very tense. But you have to, when they're saying number, you have to multiply it with the total. Okay, moving on to question number 9. So again, this is a question from your allelic frequency. So you get a lot of questions from allelic frequency, genotypic frequency, questions from based on your Hardy-Weinberg equilibrium. And from the deviation part, you get questions from mutation, from your inbreeding. Okay, concept of inbreeding. And there are numericals based on natural selection also, as well as your migration. But so far they haven't asked for any numerical based question from natural selection and migration. But it's always better to prepare yourself for any kind of question. That is what I tell my students. There's no harm in learning. And those concepts are not very difficult as well. Okay, like how the mutation part was very simple, how your inbreeding is simple, questions based on numericals based on natural selection, and your mutation will migration will also be simple because the formula is simple. And yes, one more thing. Uh, One more kind of question you get from here is the effective population. Okay, you don't get a direct question from your genetic drift, but from that section you get question from effective population size. Okay. All right. Coming back to the sum. So let's see who gives me the correct answer. Okay, Arsu is saying A. Anusha is saying C. You know, in these kinds of questions, the only place where you can go wrong is in the calculation. Nowhere else, because the formulas are very simple. Unlike the core mathematical part, you know, mathematical questions, there are so many formulas and so many different expression of the same formula. These are very straight cut formulas. So if you go wrong, either it will be in the understanding or in the calculation. Okay, so let's solve this. So they have given you that is 600, mn is 300, nn is equals to 100. Okay, and they're asking you to find the frequency of m and the frequency of n, right? So how to find out the frequency of m? The same old formula. I'm straight away going into the calculation. 2 into 600 plus 300. 2 into the total is your 1000. Okay, so if you solve this, you will get it as 0.75. Next one, if you see frequency of n, then you will see it is coming as 2 into 100 plus 300 divided by 2 into 1000. So if you solve this, you will see it is coming as 0.25. So the correct answer here is option 1. Okay. I'm in a class. Okay. Moving to the see 2 into thousands. I told you, no, these are so straight away. Formulas are very straight. Okay, so you can go wrong only in the calculation. Be very careful about the calculations. Chalo, this is the last question. Question number 10. Malini, Anusha, both of you. Same mistake. Doesn't matter. Just cross check when you're doing the calculation. You know what? Before your exam, I will tell you when you're revising, no? Just jot down the formulas in one page as you're revising. Only the formulas from one chapter. Say unit 11, these are the formulas. Okay? Formulas based on the questions which have already come and also which has not come yet. That also. Unit 8, these are the formulas. Unit 10, these are the formula. Okay? Then what will happen is just prior to your exam, you will get a very comprehensive uh, thing. Yeah, Arju has done. Yes, ma'am, I did the same. Right. So doesn't it help, Arju? Okay, others, if you have done it, very good. If you have not, today only make sure that you do it. Okay, don't delay. All right, to the last question. So then here they have told you in several populations, each of size n equals to 20, if genetic drift results in a change in the relative frequencies of alleles. Number one, What is the rate of increase per generation in the proportion of populations in which the allele is lost or fixed? And statement B, what is the rate of decrease per generation in each allele frequency class between 0 and 1? Minded, the statements are different. In one statement, they are saying what is the rate of increase per generation. Okay, so look here. Increase or decrease, basically, they are asking for per generation in the proportion of population where the allele is getting fixed. Okay, lost or fixed. In the other case, they are saying decrease per generation in each allele frequency class. Here is the difference. Allele frequency class, and here they are asking for the proportion of population. Okay. So again, this is something which I have asked my students to remember. Two statements I have given them, and I hope you remember the two statements. Option 3. Malini is saying option 3. Okay, I'm coming to the answer. Yes, Malini, you're right. Absolutely correct. So this is the two statements. For those who already know, well and good. For those who doesn't, please note down these two statements. This will come under your inbreeding section. Okay, inbreeding or non-random mating, where you will see that yes, the allele frequency have reduced. How much it is reducing, that is given in these two statements. You don't have to go into the derivation. Not required. Just remember the two statements. So proportion of population in which allele is fixed or lost increases at a rate of 1 by 4n per generation. Each class of allele frequency between 0 and 1 decreases at a rate of 1 by 2n per generation. Now can you solve it? Option 3. So you've got it? Arzu, option 3. Very good. Others? So match, tally the statement. See, you will get which formula to use. So okay, let me solve this. For the first one, it is 1 by 4n. Okay, because see, they are asking for the proportion of population. Proportion of population they are asking. Okay, so 1 by 4n, which is equals to 1 by 4 into 20, that is 1 by 80. This equals to 0.0125. Okay. Now for the next one, this is 1 by 2n. 1 by 2 into 20, that is equals to 1 by 40, and this is equals to 0.025. Okay, understood everyone? Okay, having said this, Since we do have 5 more minutes, I have one more question for you. This is from the kin selection. Can you try this question? This will be question number 11. Sorry for the wrong pointing. This is question number 11. So they're saying the brothers A and B have the same father but different mothers. B wants A to help him, which involves both benefit B and cost C. If A incurs a cost of 30 Darwinian fitness unit in that act, under what conditions should he help B following Hamilton's rule? So, what is Hamilton's rule? Relativeness into benefit should be greater than C, isn't it? So, here they have already told you that C is 30. B you don't know, you have to find out B. What should be the R? Same father but different mother. That means what? What is A? What is the relationship between A and B? Who can tell me? What's the relationship between A and B? Come on. Okay. Yes, absolutely, Arzu. It is 0.25. But can someone tell me in words like what is the relationship between A and B? What kind of brothers are they? Are they siblings? Are they cousins? Are they stepbrother? Step brother, yes. So step brother, the relatedness is 0.25. Right? Now you solve this. Now you solve for this. Okay. So you will see, so 0.25 means how much? 1 by 4, isn't it? This one definitely is not right. B greater than 30. How much greater? Already cost is 30. Okay. So this is not right. 60, if B is greater than 60, if I take 60 also, it will be less than 30. Right? Only if I take B as your 120, how much you are getting? 30. Okay? So B has to be greater than 120. So 120 is giving me equal. This R and B is becoming equal to C when I have 120. So the minimum value is it should be greater than 120, only then I will be able to satisfy my Hamilton's rule. Okay, this is a minimum value you have to find out. So this is all for today's session. I hope you have found this interesting. And this has cleared many of your doubts, because the entire reason for taking only the mathematical part from your unit 11 is to give you a clear idea as to how to solve them and also make you practice. So, for how many of you this has been a very helpful session? Okay, thank you. Okay, so this is the end of my discussion with you all. So, I'll take this opportunity to wish you all the best for your upcoming exam. Okay, analyze the question carefully and then answer. Don't worry, you can always change your answer. Okay, this is the computer based thing. So until and unless you hit the submit button, you can, till then you can change any of your answer. Okay, so analyze, cross check, and then give the answer. So with this, thank you everyone. Malini, I don't know about that. Let's see what they say. Okay. All right. So with this, I will end the session. Thank you.